cancer research. And Dr. Barham is the director of the National Cancer Institute. Asked him to chair a very important panel at the Institute of Medicine, as part of the National Academy of Sciences, to envision the future for the appropriate use of omics technologies in the research continuum from basic out through applied and out into actual use with patients and participants and things like that. And so that's, I think, where this regulatory science comes in. I want to hear that part. You know, his research interests are exciting, and he's been working closely with Raji Menon in a whole worldwide consortium with the Human Proteome Organization, which you're going to hear about. And his interest involved, you know, this very interesting area of protein splicing, which is clear that this is a critical part of how proteins get their diversity, especially expressing themselves at the level of the tissue. So I can't say anything more, but at least I can say that. Gil, we're very proud to have you with us, and we're happy that you're here today to give your talk, Splice Variants Proteins as a new class of cancer biomarker candidates, more complexity for regulatory science. Thank you for coming to our seminar series of doing this.
Okay. Well, thank you, Brian. We're a team.
We are.
Okay. This is a new topic, basically. Some of you have heard Raji or me on this subject before, but I want to put it in broad perspective. And I think in the coming years, all of you will need to take account of splice variants at the transcript level and the protein level in understanding biology, and if you're working on such applications, developing biomarkers for diagnosis, for prognosis, and for choice of therapy. Hope most of you are familiar with this scheme that Brian and I created a few years ago. It was at the heart of our integrative biomedical informatics for our national center. And the notion is you can start from the genome and work your way to the phenotype or phenome of the individual or the species, or you can start from the phenotypic observations and work your way back through all the metabolism, physiology, to the omics level. And this chart, which over here points to post-translational modifications, transcript variants, and other modifications, captures some of the complexity at the omics level. For all lectures, University of Michigan, you're required to have a disclosure if you have any relevant outside activities. I don't know that they're relevant, or I don't consider them to be potential conflicts for this talk, but you are entitled to know that I'm on the board of directors for many, many years of Amgen Inc., a biotech company, and then a small company called Etubix in cancer gene therapy and immunotherapy. And recently, this company started by Rahul Chanayan and Xiaomen Wang, called OncoFusion Technologies, which I think is very promising. Anyway, the outline of today's talk is the context of omics-based predictive tests and targets, and then strategies for cancer biomarker discovery, with a specific emphasis on the Splice Isoforms. And then, if we have time, two related topics I think are quite interesting. One is the bridging of our proteomics work with the structural biology work of Yang Zhang and his whole group, and finally the Human Proteome Project, global project that Brian mentioned. Here's the report. It's called Evolution of Translational Omics, Lessons Learned and Path Forward. It was released in the end of March 2012. How many of you have seen this report? I heard about it before. Very few. Well, I'm glad I put it in. So the goals of this report were, number one, to define best practices, having started with less than a best practice, Brian mentioned, for discovery and translation of an omics-based test into clinical utility, clinical trials, and clinical practice. So that meant that we had to cover discovery phase, confirmation of findings, a step routinely omitted, lockdown of the parameters, analytical validation, and clinical utility and its validation. And then part two of the report was to recommend actions for different parties to take. And this covered the investigators, their institutions, very importantly, the funding agencies, the journals, and the Food and Drug Administration. There's Shira. Anyway, I gave a talk about this same subject at the FDA a week ago. And these are very practically minded, practically oriented people who are quite excited to learn about what's coming their way. This is the story of the report in one slide. We could spend a whole lecture on this. I just want to point out that we deal with this must be a pointer. Yeah. The discovery phase, the test validation phase through to clear laboratory in most cases. But in this phase, you must lock down the laboratory assay, you must lock down the computational procedures, and you may not use any of the samples from the training set as part of the test set. If you see a paper with a violation of that rule, it should be discarded. And then there's the evaluation for clinical utility by several different approaches, 3 different approaches. It is highly recommended to contact the FDA for consultation, which they are very helpful about, or, and it's going to be used to actually make a difference in the care of the patient to get FDA clearance or approval.
Was there a recommendation for the laboratory developed tests that are not currently under?
Yeah, this is a very important but subtle point, complex point, that there are 2 paths. You can have a lab-directed test, which means it's performed only in the lab that developed the test. Can't be exported to another lab, and they can do it if they're capable enough, and the FDA doesn't object. The preferred route is to take it to the FDA and have actual FDA formal clearance, equivalent of approval, and usually that actually leads to a more robust result. However, there are tests right on this list of the case studies we presented that did not go through the FDA, but instead got, in most cases, a very rigorous review before Blue Cross Blue Shield or others would pay for the performance of the test. That's another opportunity to look at the quality of the evidence. Anyway, there's the Duke molecular signatures, and here's an example of a paper from the Duke team. Does anybody see the key word on this page? Retracted. Big story. Oops. And the lessons learned are here. You have to have a well-designed plan. It's good for research of all kinds. Let's say protocols and documentation of everything that's done. You must make the data and the code fully available. Nothing less will be tolerated in the future. It shouldn't be tolerated by journals now. There must be separate discovery, confirmation, and validation sets of specimens and analyses. Lockdown, as I described. I highlighted here interaction with the FDA, oversight of the assessment of specific clinical utility, not just any old clinical study to match up for a specific use. And finally, oversight by the co-investigators. It's not good enough to say, I just put my name on the paper, I have no idea what was in the paper. That word retraction appears on the CV of every single one of the many co-authors. 162 co-authors on 40 papers, of which 27 so far are retracted. And their responsibilities for the institution. Okay, that is the context in which the development of omics-based tests, omics-based pathways and networks analyses that we're going to talk about next, can be channeled to practical medical application. Here is a story about DNA, RNA, and protein. We start with the Human Genome Project, which was 1986, led to these 2 full-issue, iconic issues of Nature and Science almost 13 years ago. But you know what happens when there's a big news item? A few days later, it's like, been there, done that, what's new, right? Well, what's new is shown here. What do you see? Where's the DNA? The double helix. In fact, somebody told me that the DNA was trying to kick the protein. But in fact, if you follow, there's the right hand, right? And here's the left hand of the protein actually pushing the DNA off stage, shoved off stage, Brian says. And here is the message, the real stuff of life, racing to decipher the human protein set. And that's what's been going on. And now we actually have very substantial progress and a plan to try to do the entire set. We'll talk about that at the end. So, proteins are the major action molecules of cells. Proteins and their isoforms are highly dynamic, means they change many times, many-fold in concentration. Play critical roles in gene regulation and among their many functions. The advances in the last decade on instruments, reagents, and bioinformatics and now facilitate integration and modeling of data from multiple omics platforms. This is our business. And finally, proteins are the primary targets of most drugs, and nowadays are often drugs themselves, designed to hit specific targets as well as biomarkers. So these are 4 different ways of going from a tumor, if you're trying to do cancer proteomics, to biomarkers in tissue and plasma. We've learned it's actually quite hard to start from the plasma. It's a very complex sample. It's dominated by one protein called anybody? What's the most dominant protein in plasma? Albumin. Constitutes 50% of the plasma protein by weight. Another 21 proteins, lipoproteins and others, plus albumin, account for 99% of the protein mass. And they have many different isoforms. So what you measure mostly without really high-resolution techniques is over and over the same abundant proteins and their isoforms. It's not good enough to find biology that is secreted from or released from tissues throughout the body. But you can start with microarray or other RNA-level studies, or maybe even gene expression studies, gene studies. And one of the great examples here is TMPRSS2, work from the Will Chinayan's lab with our student, Dan Rhodes, and another MD PhD student, Scott Tomlins, that transformed thinking about prostate cancers. Always speak of cancers in the plural, through this gene fusion mechanism. Also, metabolomics revealed a key role of the metabolite sarcosine, prostate cancer. So you can take that information and the enzymes, pathways involved, and go and look in the circulation or in urine or the blood to see if you can find what you discovered in the tissue. You can also start with tissue studies and perform SRM, that's selected reaction monitoring, looking for proteotypic peptides, peptides unique to a particular protein, and see if you can find that peptide in the circulation. If so, you can quantitate it, track it, health and disease. And in fact, there is now a great result of the last 2-3 years, a set of SRM peptides and synthetic peptides that can be labeled to do quantitation, and the associated mass spectra from mass spectrometry corresponding to all the 20,000 gene-coded proteins in humans, and similar for the mouse. That is reagent availability for planning experiments. You can also go to the peptide atlas databases and see if the peptide you are specifically interested to measure has previously been detected. If it has been detected, you have a lot higher confidence that you could also detect it in similar specimens. Another approach might be, say, well, why don't we do PCR for proteins? Anybody know how to do PCR for proteins? Nobody? Could win a big prize if you could. We don't really have a way to do that. But what we do is have nature's way. And nature's way to make amplification of a protein signal is number 3. The immune system. Okay? Certain amount of protein or protein fragments from a tumor, maybe a tumor antigen molecule, gets out into circulation, and it gets into the lymph, and it stimulates the lymph nodes to produce antibody against that sequence. You can actually measure the antibodies, and they're typically 1,000-fold higher in molecular concentration than the antigens for which they were raised. Finally, however, and the topic of today's talk is number 4, which is to identify alternative splice isoforms of biologically meaningful proteins in cancers, measuring in the cancers, in the plasma, humans or mouse models, or in cell lines from cancers. And this is exactly what we're working on. Well, blood is plasma plus cells, red cells, white cells, platelets, basically. Yes, but the reason he does that is there are also exosomes and mirrors and so forth that you might detect and are being detected. So it is a broader category, but in fact what is almost always measured is plasma or serum. Serum is plasma that's been coagulated, formed into a clot, and all the clotting factors are consumed. So, how many of you have read about splice variants in basic biochemistry? Not many. Maybe they're just short arms. Okay. Here's the story. Bacteria don't have splicing. Yeast, which in many ways are like little humans, or little cancer cells anyway, have very limited splicing. But multicellular organisms have increasing splicing involving multi-exon genes. So you know, genes now, we understand, have exon intervening sequence called intron, exon, intron, exon, intron, and ends. So I'll show you formulas in a second. In the early literature, there was a special finding to say that one additional or several additional splice variants were found for a particular transcript or protein. Now, nearly all the multi-exon genes are found to have multiple forms, splice isoforms. The implications of this are actually huge. We still talk about and see thousands and thousands of articles about up or down expression of genes and their proteins, genes, transcripts, proteins. But in fact, those are all mixtures of these gene products. And the assumption that you can add up the number of molecules of different isoforms and call that the concentration of the protein is biologically misleading, because it's been shown many, many examples now that the functions of the different splice variants of the same gene can be quite different, quite opposite, like pro-apoptotic, anti-apoptotic, activating or repressing transcription, etc. So we have many examples of this, and the need is to identify the structure and function relationships of each variant, and that's going to become essential. Also, with modern technology, as I'll come to in a few minutes, let's say RNA sequencing, deep RNA sequencing, the different splice transcripts are laid before you, and they're constantly a relative number, concentration. So it's pretty hard to say you can't analyze something that you can't see, because now it's shown to you in the analysis. Okay, let's move a little faster on this. Here's a picture of this exon-intron scheme. Exon, intron, exon, intron, exon. And the introns have to be spliced out to make the mRNA. Previous versions called the pre-mRNA or the heterogeneous nuclear RNA. And it doesn't happen simply. This is an incredible process that results from evolution. There's a molecular process and a big complex of proteins and ribonuclear proteins called the spliceosome, described here. It's actually a very big subject, and it's been studied carefully to show how the intron sequence is organized and how it's spliced out using those markers and the different features Hopefully some of you heard about this in biochemistry. The point is that there's a new class of molecules, biomarker candidates. This alternative splicing generates protein diversity, as Brian said, without increasing genome size. In fact, there are other species that have more protein-coding genes than we do. That was quite insulting at one time. But now we have all these splice variants to boost our egos. And there are some dramatic examples, not just in cancers that I'm going to talk about, but in neurosignaling. In fact, in the early years, this is from a review article 12 years ago, for these three tremendous examples of extreme combinatorial molecular features that underlie the hearing process in the cochlea hair cells, synaptogenesis in neurons, and guidance and movement of axons through the brain to their targets, yielding literally tens of thousands of mRNA isoforms with distinct receptors and cues. In this case, early work in Drosophila, but now shown in many other organisms. Now, the mechanisms are multiple. There are different splicing events. And one of the things we're interested about going forward, something Roger and I have already suggested, is that we might be able to analyze the kinds of splicing events that are associated with particular changes in proteins. This is yet to be done. Nobody's done this to my knowledge. But here you can see perhaps the easiest example is right here, mutually exclusive exons. This is called an exon swap. You either use this exon or this exon, and never both. Or you can have a cassette. It's either in or removed, in which case you skip an exon. Or, as you'll see from some of the examples, you can retain the intron. And on the polysome, protein synthesis will march right through. If it is in frame, it will just be an extension or expansion of the protein sequence. If it's out of frame, it will probably truncate the protein. And other models there. And we have examples from our data of all of these. So here's the scheme that Roger Menon and David States and others of us were involved in starting. Building a database from Ensemble for transcripts and from ECGene, which is based on cDNA, to predict all the potential protein sequences that could occur and to include those that had never yet been observed. So we'll show how this plays out. This modified ECGene database, we call it, has exactly this scheme. All this is described in cancer research in 2009 and for pancreatic cancer studies, and the following year for breast cancer. I'll show you those references now. So these are molecular models in which genetically modified mice have been created with exactly the molecular lesions found in humans. Almost all cases of pancreatic adenocarcinoma, ductal adenocarcinoma, and 15% subgroup, that's the ERB2, HER2-neu subgroup of breast cancers. We've also been looking here at the bottom at multiple cancer cell lines. We'll give you examples of that too. So here's the pancreatic cancer story. Altogether, we found 420 alternative splice variants. 328 were already known. They were in the Ensemble Swiss Prot databases. The others we called novel. No one had ever reported them before. Of course, they're really equivalent because by our next study, we reported them and they were now in the known category of splice variants, and some of the ones we found for pancreatic were also found with breast cancer. Quantification here occurred through a special isotopic labeling method, which limited the number of proteins which you could actually label. They had to have a cysteine in the particular peptide in order to be identified and quantified in this particular case. Now, what are examples? This is out of those 420 splice variants. Let me show you 4. This is a splice variant of the interferon receptor. It has 243 amino acids, this variant protein. It's identified by a unique peptide, doesn't occur in any other protein. It was found from 70 different spectra. This is an extremely abundant protein and extremely confident identification. It's upregulated, higher expressed, this form of the interferon receptor in cancers, and it's much shorter than the normal protein, which is 513 amino acids, so more than twice as long. The result is that there are missing motifs, missing domains of this protein with the CAMP and cyclic GMP-dependent protein kinase phosphorylation sites specifically affected, removed. The intensity and distribution of these interferon receptors have been used to predict the response to therapy with interferons in pancreatic cancer patients. So we think this is something that really needs to be understood at the isoform level, not just overall protein. Here's another example, lipocalin. We'll come back to this in terms of human translation. Another example of a shorter variant, 200 amino acids versus the full length 284, identified by a unique peptide, 15 different spectra, distinct spectra. Again, upregulated in the tumor versus normal. In fact, we weren't measuring the tumor, we were measuring the plasma from mice with tumor. Okay, so we're actually detecting these peptides and these proteins and digesting them to peptides we could detect in the plasma. And lipocalins have been reported to be involved in a variety of cancers. And we've also found it in our human translations. I'll show you in a minute. Two more examples. This is malate dehydrogenase. This is an example of an intron read-through. The peptide that was found here is normally not expressed. But in the splicing, an intron was not removed. And it was possible to read through that region in the mRNA and generate additional sequence in the protein. Again, increased in expression in the tumor sample by 5-fold. And this is an important step in citric acid cycle, Krebs cycle, and gluconeogenesis, so energy metabolism in cancers. And here's the critical step in cancer energy metabolism, which is pyruvate kinase, M2. This had not been found before in the mouse. Well known in the human. It is, in fact, the switch that turns glycolysis to aerobic glycolysis, and it's called the Warburg effect, described in 1929, and now understood. I'll show you beautiful pictures from Yang Zhang's group and us on exactly what happens. So you could say, what's the possibility these are mistakes? There's a risk that you get the sequence wrong when you match sequences to databases. So the method here was to take a sample of the tumor from the mice whose plasmas were studied and prepare the mRNA to prepare QRT-PCR assays, which Raji did, and then see if you can find the mRNA that matches the sequence that you deduced for the protein from that mRNA. And in each case, we were able to do exactly that. Now, this is a whole big story, but the short version is that Sam Hanash's group, having moved to Seattle, had provided us the data set for this pancreatic model. And then they took the same data, all these proteins that were identified, including the ones that had splice variants, in the mouse model, and predicted which ones might be upregulated in humans by homology, and then studied human samples. And the human samples they studied were from my old study, specimen bank, for the beta-carotene and retinal efficacy trial, trying to prevent lung cancers in men and women at high risk for lung cancer from heavy smoking, long-time smoking, even among former smokers, and from asbestos exposures on the job. And of the 9 that they were able to assay by ELISA assays after all this big funnel of potential protein candidates, 3 of the 9 were among our 420 with the splice variants, including that lipocalin that I showed you a few minutes ago. These were found before the diagnosis of the cancer. These were pre-diagnostic specimens from the chemoprevention phase of people high risk who didn't have any cancer. But some of them, out of 18,314 participants in the trial, developed lung cancers and other cancers as they were followed up. So that's the pancreatic story. And Raji and I are talking now with Diane Simeone, head of pancreatic cancer research here, about doing studies to see what we can predict might be found in actual human pancreatic cancer patients. Next story is breast cancer model. And this is really quite a lovely paper from a totally different group at the Fred Hutch. Their data were available in databases. In fact, the database CRANCE that Phil Andrews established here with the two of our students over the years. And it's notable for their finding many proteins. Tight cutoff, 158 proteins, higher in a tumor than in a normal breast mammary tissue. They said, well, let's measure these by antibodies. How many of you tried to work with antibodies? Okay. So they were able to find and purchase antibodies against 24 of the 158, only 24. And of the 24, only 7 gave results that confirmed the claimed specificity. And of the 7 that reacted by Western blot, only 1 worked by ELISA. And that 1 was one for which there was already plenty of evidence that it was a pretty good marker. So no new marker was found. However, they went on to use the SRM, the selected reaction monitoring, mass spectrometry targeted proteomics method. And they were able to find a couple dozen that became quite good candidates, including things that we're interested in. In this case, remember we had 420 total splice variants for the pancreatic. Here we had 68 novel, 540 known, 608 splice variant proteins in the HER2 breast cancer. And in this case, we could quantify all of them because we did it by spectral counting. We didn't have a mixture of the labeled and unlabeled versions, so we could actually count the spectra separately for the tumor tissue and the normal tissue. Here we picked 31 of those 608 to see if we could demonstrate the mRNA by QRT-PCR, and essentially all were found. 29 had a high correlation in the right vector. Let's say where it was up, it was up, it was down, it was down, both for the protein and the mRNA. A couple of interesting examples here out of 608. And of course, we try to annotate as many as possible. This is a story of a gene called ROGD. It's a zinc zipper protein, zinc finger protein. And here again, the peptide was from the intronic region that was retained. And what's fascinating about this is that there's a phosphopeptide motif that emerges which directly seems to interact with the C-terminal domain of the breast cancer 1 gene. Wow, that sets off bells, because breast cancer 1 is a very important mechanism for a certain subset of breast cancers. And lo and behold, we found another peptide which mapped to a protein, a transcription factor protein, SOC7, again in an intronic region, and again introducing this functional motif of this phosphopeptide site that maps to interaction with the C-terminal part of BRCA1. I've been trying to persuade a guy over in BSRB who works on breast cancer genes to see if he could actually study this at the protein level, and maybe someday it will be done. Well, it's not the peptide, it's the protein, but we detect the proteins after cryptic digestion through the peptides. Right. And it's the splice variant that has this additional feature, which is what binds, in this case, the transcription factor to this site. Here's another one. This is one that came out of that SRM study I just told you about. Our colleagues who provided the data set here. And by the way, we offered them to be co-authors on the paper because they were quite cooperative about their study. But they said we didn't really do anything. Our data are available for anybody to use. We're thrilled to use it and cite our work. I'm happy to cite their work and acknowledge them appropriately. Okay, so here's this fibulin. This is a well-known marker in breast cancers. This is just like one of the examples I gave you before, where the shorter variant is lacking a cyclic AMP, cyclic GMP-dependent protein kinase phosphorylation site. And that interferes with the inhibition of cell proliferation. Therefore, there's more cell proliferation, more cancer growth. And it's upregulated. This form is upregulated in breast cancers. This is a little hard to read, only to remind me to say, as I told you earlier, that each one of these has an interesting specific mechanism of splicing being altered, including the intron retention, different frame, 5' UTR, exon skipping, all the different mechanisms found. And we put this through MIMI, NCIBI is MIMI, plugged into Cytoscape, and can map the protein-protein interactions. All the ones in bold that you can't read too easily are actually the parent gene symbols of alternative splice variants found only in the tumor sample, not in the normal tissue. And there are some very interesting connections in those pathways. Yeah. In this case, it was a tumor assay. In the first example I gave you, we didn't actually assay the tumor for protein. We assayed the protein in the circulation, in the plasma, in tumor-bearing mice versus non-tumor-bearing mice. And in fact, that's exactly what we want to do. Just like Lee Hood was saying 2 days ago, we'd like to be able to use the blood plasma, as a window on what's happening in organs or tumors or other sites in the body that are too hard to assay, let alone assay repeatedly. Okay, now something big has happened. I already told you this maybe 20 minutes ago, which is that instead of starting from mass spectrometry of the proteins, we can start from the deep sequencing of the RNAs, find the transcripts. We still have to answer the critical question whether those transcripts are translated into protein. And if they are, what do they do? What is the different folding, conformation, structure, and thereby the different function of the splice variant compared to the canonical or normal protein. So here's a paper from our big Human Proteome Project team on chromosome 17. How did we choose chromosome 17? Well, our Boston colleague, Bill Hancock, proposed it because it is the home of the gene for HER2/neu, ERB2. And we said, this is our special interest, ERB2 breast cancers, and his. So we came at it from very complementary points of view. For the RNA sequencing, Mike Snyder, big figure at Stanford, is our partner, and a lot of people involved in this project. Everybody's played a key role. So one of the first things I did was to have Chris Gates from Oncomine, let's say, Compendia Biosciences, who was taking courses here as a master's student, come by and help me ask some questions in the Oncomine cancer transcription database. First question I wanted to know was, how many genes on our chromosome 17 are overexpressed or differentially expressed in ERB2 type of breast cancer? And it turns out that there's a whole bunch. These are just the top 20 that are overexpressed, that are coded for on chromosome 17. Now, some of these are way down the list, but you can look at those first ones. 1, 3, 4, 5, 5. That means at least 5 of the top 6 are on this chromosome. I compared HER2/neu type breast cancer with estrogen and progesterone receptor positive breast cancer with triple negative breast cancers for the genes on chromosome 17 associated with that particular subtype of breast cancer, and there was no overlap at all. These are 3 entirely separate top 20 lists. These are different diseases, happen to be called breast cancers. Here is a different question. This question asks, of the top 20 genes for HER2/neu breast cancer in the entire genome, how many are on chromosome 17? And the answer is 13 out of the 20. That is big association. And you can see the scheme, and there's our 1.5, 2.5, 4, 5, 5. So, top about 5 or 6 in the whole genome are those ones on chromosome 17. Well, we can map these, and we actually have a lot of information in a figure like this. Here is ERB2, HER2 new. And the code here shows we show what kinds of post-translational modifications they have. Phosphate groups on serine threonine or on tyrosine, acetylated, glycosylated on nitrogen, glycosylated on oxygen. These are very important post-translational modifications. And we also know over here if there's evidence of splice variants. So we can capture in this kind of a scheme a lot of information that can then be annotated further. Please pay attention. Here's ERB2. The chain comes down this way. Here's GRB7. We're going to see that again. And up this way, here's CDK12 and FBXL20. I'll show you those in a slide in a couple minutes. Yeah. Exactly. Okay. So it's like this. Okay. And this is exactly the linear order on the chromosome. The other thing maybe to say in advance is that these are somehow under important co-regulation. Maybe I should say this. When we started the Human Genome Project, chromosome by chromosome, as well as disease and biology processes, some people, a lot of people actually said, that's dumb. Protein pathways outside of bacterial operons are from genes anywhere in the genome. I mean, proteins aren't, genes aren't lined up together in the vast majority of cases. Well, maybe in the majority of cases that's true. But here's a situation where essentially all of these can be activated, overexpressed together in what's called an amplicon. And this is a whole nother question of how are amplicons regulated? And they're highly variable. It can be as few as, I mean, it might be just ERB2 by itself, ERB2 plus 1 or plus 2, or as many as 23 have been detected to be coordinately upregulated and expressed. And they come together in pathways. This is not random stuff. It is fascinating biology in cis position, cis regulation within a given chromosomal segment. And you know, and I don't see Indika here, but he and Brian and others are interested about, something about that segment of the chromosome being massively upregulated somehow together. Very interesting. So here's the amplicon, as I just said. 2 up to 23 adjacent genes co-expressed. Many, but not all of them, have been observed in our studies to be overexpressed as transcripts and as proteins. And we can annotate their functions to things that are highly relevant to cancer invasion, cancer migration, and eventually metastasis. So here's a dramatic example. This is the shorter splice variant transcript we call SPT for ERB2. 6 different ERB2 cancer cell lines, 2 from breast, 2 from colon, 2 from gastric. The 149 and 190 are the breast cancers. And their expression of this splice variant is extremely variable, especially high in this one, which is the amplicon high expression cell line for breast cancer. The 149 has only normal low-level expression of ERB2, and instead has much higher expression of EGFR pathway. And in fact, EGFR has another name, which is ERB1. And ERB1 and ERB2 can form heterodimers. The biology here, we're looking into these pathways in great details. I'll give you a clue later. Okay, here are those 3 proteins I showed you from the sequence picture. So here's GRB7, which is 2 over from ERB2. Here's CDK12, FBXL20, and they're differentially expressed in different cell lines. But again, this SUM should be 190, has this huge overexpression of GRB7. This the same one had such high expression of that short transcript of our ERB2. And I don't have experimental evidence, but I'd be willing to bet my nearly last dollar that that short transcript of ERB2 and this isoform of GRB7 are specifically co-expressed. It's not just GRB7, any form of that protein or that gene. The same for the ERB2. Here's yet another example. This is septin-9. And there's 7 different splice variants here. They're listed up above of different lengths. And they're shown very different patterns in these different cell lines. It is no longer acceptable to just speak of septin-9 as a mixture, undefined, ignoring all the complexity. And this is a summary of many, many splice variant transcripts and proteins through the chromosome 17 analysis. Too much to cover right now. Also, we've done network mapping. We've used concept gen from Maureen and David and Genomatics. And we can map certain kinds of pathways. The most interesting little piece here is the last item, I think, that there are many splice variants which have the same transcription factor recognition element in the promoter sequence. And that's very likely to mean that that's a common mechanism for turning on these multiple genes coordinately. Our latest paper, just recently submitted and awaiting final acceptance, is for the 2014 January special issue of Journal Proteome Research. This is a distinct splice variants and pathway enrichment in cell line models of these breast cancer cell types. So again, the same 3 breast cancer cell lines we've been talking about before at the top. Their features, 2 of these are inflammatory breast cancer with opposite pathways activated. That's the ERB2 or ERB1. And then there's an adenocarcinoma, which also has high expression of ERB2. So we have certain comparisons here which are worth a lot of analysis. Given the time, I'm going to go quickly here. But just to say that the patterns are quite different, but the ones that are overexpressing ERB2 are much more similar to each other than to the third. Here is an example of differential expression for the ERB2 way up at the top. And what we do is use the I-TASSER method to look at the actual splice variants and try to see what happens to the folding of the protein and what we might predict about its activity using those methods. Here's the same for the ERB1 EGF receptor. Not the same, but parallel analysis. And these are pathway analyses for ERB2, for caspase, for ubiquitins, and all of these show marked differences across these 3 cell lines, but greater similarity for the ones that share the upregulated pathway than for the ones that do not. So there's real biology there, and this is the quantification of the transcript abundance. And you can see at the top, positive numbers green for the EGFR associated proteins, and red, negative numbers down here for the ERB2 associated proteins, and those show up in the pathway analysis and in the functional analysis of those pathways. These are the pathways. It's really hard to see this, but the green are the ones that are highly abundant transcripts, and it turns out that for this, the ERB2 signaling, the associated steps go very differently from the ERB1. And we have a whole big section in this paper about the details. Not only that, but things we didn't anticipate, like the role of MYC up here. MYC up here. See, and the size of the circle, the black has a certain. Black shows that we actually found it in the proteomic analysis as well as in the transcripts, because transcripts are much more complete than the proteomics. And here's a family member, GRB, not number 7, but number 2. Here's GRB7, our friend. And these start to look real familiar after a while. And the differences are also very striking. Over here, we pull in the STAT pathways. So that's quite a distance. The ones that have the shortest lines are the closest. You see this short line here? Here. So this is nodes and edges analysis. Sort of. Yeah. Okay, this is redundant. Beware generalized statements about up and down regulation of protein expression. Recognize these splice variants. Figure out how to analyze them and use splice variants and PTMs to figure out structure-function relationships. That leads me right to this third topic, which is this really, I think, significant paper with Yang's group. And by the way, in 2 hours, we'll be celebrating the Dean's Award for Yang Zhang as the honoree for this year for all the middle and senior level faculty of this medical school for the distinguished research and his record. And we're very proud of you for earning that distinction. Hey. Okay, so this is quite a nice story. Very briefly, if you walk down our corridor eastward and you look left, you could get a full education about I-TASSER. Right? Anybody seen these reprints hanging? Take a look. They're up on the walls on the second floor for a reason. Part of the educational process. Anyhow, this is a very novel and now extremely widely used threading algorithm approach to protein folding, protein conformation, and well, the story was this. One day I walked into Yang's office there, and I said, I've got a real interesting problem I'd like to try out on you. This is what you should do all the time. You know, in big university, all kinds of people with great expertise. Take your questions to people. So I said, have you ever tried to use your methods to distinguish the folding and structure, potential function of 2 splice variants? He said, no. I said, well, are you aware of anybody else who has? No. Do you think it could be done? Maybe. Would you be willing to try? Yes, of course. But, you know, we're very busy. We've got a lot of projects going on, so like anybody might say. He said, well, maybe we could do the first analysis for you. Would it be satisfactory if I gave you the results in, say, 48 hours? I said, yeah, that would be quite satisfactory. In fact, I am so keen to see this result, I could stand right here and wait 48 hours. So in 48 hours, we had the predicted structures for this, what I would call positive control. This is that pyruvate kinase M2 I mentioned before. That is the switch to aerobic glycolysis based upon using exon 9 or exon 10 here in this part of the molecule. It's the fructose bisphosphate binding site. And they fit almost exactly. But it turns out there's a difference up here. This is the ATP binding site, and it affects the phosphorylation of this specific tyrosine, which is critical to the switch in cells to aerobic glycolysis. This is work from Lewis Kentley in Boston and X-ray diffraction colleagues. So this is our positive control, where the structures of both forms were known, and we could see how well I-TASSER could detect the similarities and differences. So then we did a bunch of other studies. Specifically, in the huge protein data bank, 80,000 protein structures, or partial protein structures, there are only 7 proteins which both of a pair of splice isoforms had full structures. So, the team ran all of those and got excellent fit to the known structures. Then we looked at 3 pairs which were not structured, but related to cancers. They have opposite functions. BCL-X, caspase-3, skipped related to OSR2. And this is a transcription activator versus repressor. Both of these affect apoptosis. And there were striking differences in domains different between one and the other splice variant that are good candidates to explain their differences. It doesn't prove everything, but it's very nice information to guide what could be more laboratory analyses. Then, and here are the pictures that go with those. It's a little late to go through all that. But these are, the thing to look for here is that red and green are the 2 different molecules. And when they fit exactly together, you get the red-green overlap, like you can see very nicely in these pictures. But where there's a missing, see where the arrow is down here for this OSR2? There's just nothing from the other isoform. And same thing in this one, in all three of them actually. So we chose 5 protein pairs from our HER2/neu mouse model, breast cancer. And we had these criteria, and they were interesting proteins because of their biological functions. The splice event we could identify, and we could show what really happened between the two. In the case of annexin 6, for example, which was reported to undergo phosphorylation as part of more rapid proliferation of the cancer, there was a shift which removed 6 amino acids in the sequence, 535 to 525 to 530. The result was to move all the rest of the molecule 6 residues toward the N-terminus. And this affects the exposure of serine residues for phosphorylation. This is a model picture. The area that's under discussion is right here. You see the red and the green together, and then here you see just a big glob of green for those residues, not matched by the red. And we later did, for a subsequent paper, a further analysis with the space-filling models, and you can see here tyrosine and serine that could be excellent candidates for phosphorylation. Over here, it's just this, the serine moved 6 positions to the left. It's still available. And we'd love to see this experimentally characterized. But we think we're onto something important. It's not just our own work here. There is a group that reported a year ago about BRCA1, which has multiple splice variants. One of them has a missing, let's say, deletion of exons 14 and 15 of this very large protein. And it turns out that that deletion has decreased DNA double-strand repair, impaired genome stability, and it matches a phenotype without knowing everything about how it functions. So I hope these examples will drive a lot more work in this area. The things that can be done, and we hope to do here, are to devise SRM assays for the splice variant peptides of the most striking proteins from our pathways analysis and see if we can find them in patients with the particular type of cancer. We'd like to translate the specific findings from mouse models and human cancer cell lines to patients. This is translation. And in general, we'd like to experimentally test the predictions from the I-TASSER analyses, starting with this annexin 6 that I just showed you the models. And thanks to everybody. I think I mentioned nearly everybody as we went along. Now, just a few quick slides on the Human Proteome Project. This was announced in 2010. In the past year, we've had 48 publications. I'm not on all of them, or even most. But we had a special issue of the Journal of Proteome Research in January and again in June. We're working on another special issue with multiple papers for this January. The goals are to build the parts list. Some of you heard Lee Hood say yesterday about the Human Genome Project being a parts list. And we'd like to identify a protein product, at least one protein product, from each of those 20,000 protein coding genes. And then the variants, isoforms, from post-translational modifications, from splicing, from mutations, polymorphisms. Second, and this is a really important part of this project, is to make the proteomics accessible, robust, useful to a very much broader array of scientists. You hear about transcriptome analysis all the time, more and more stuff in metabolomics, of course, genome analysis, but that critical step of the proteins and the protein isoforms, which cannot be predicted from the genomic sequence, must be analyzed. And these new high-throughput instruments, the medium sophistication instruments that would be good for clinical labs and desktop models for labs that aren't primarily proteomics but can use it. The knowledge bases for identifying and characterizing these proteins and their pathways and networks. This is what we want to make available broadly and show how they can be used. So this project starts with mass spectrometry, antibody capture of proteins in a knowledge base. Then we have this adopt a chromosome approach, teams all over the world, one chromosome each. And the biology and disease-driven projects is another way of showing the same thing, chromosome-centric, biology-centric. And these are the team leaders for each chromosome. So you just look around and you see all the countries. And here's our chromosome 17 up here, so forth. You want to see what it looks like on a map? Here's a map. And it's pretty interesting. The biology and disease-driven started with projects, including one I led for 10 years on plasma, up the top. Kidney and urine, and we just submitted a paper comparing kidney peptide atlas, urine peptide atlas, and plasma protein atlas for which proteins are in common and maybe why, and which one's not. Cardiovascular, liver, brain, stem cells, and then diseases. So the organ-specific diseases, glycoproteomics approaches, cancers, diabetes, infectious diseases. A new project on the eye. The eye is a very complex organ, 8 different structures. And we combine the organ-specific and biofluid specific and technology-based initiatives that we developed over the last 10 years into this biology and disease-driven part of the HPP and added a few new ones, and there are others asking to be included as well. I already told you this key point about the rationale for the chromosome-based approach. The proteins are not generally coded by genes on the same chromosome, but we recognize a biological rationale consists of regulatory phenomena and co-expressed genes and amplicons. Example from chromosome 17, and there are others. There's also a fascinating result in the manuscript for next January special issue from teams in China. There are so-called deserts where there are genes but no transcripts found, no proteins found, and the genes annotate to produce defensins. These are smallish proteins, really polypeptides, that have antimicrobial activity, and it looks like they are only expressed with inflammation or infection, maybe specific infection. So the question is, for the proteins that we have not been able to detect yet, is it because there's no transcript, in which case there won't be a protein in that tissue of study? Or is it the level is too low? Or it's unstable? Or maybe it's only expressed under certain conditions, maybe very early in life, or maybe under disease condition, stress like infection and inflammation I just mentioned. So we're really dedicated to finding these missing proteins. Here are articles about these projects. Here's the big special issue. Altogether, there were 22 articles from the HPP teams themselves, and another 11 from other people. This is our second goal, is to get other people to join in who turned in manuscripts that were good about membrane protein fractions, about other interesting challenges that become part of the HPP special issues. And we've built a master table of the data sets that we utilize that have high credibility and very low false positives. And these are NextPro, Peptide Atlas, and GPMDB for mass spectrometry information, Human Protein Atlas, antibody-based protein detection and tissue expression. And here we are, 2012, 2013 January special issue. We had about 13,000, so 6,000 to 7,000 missing of the 20,000. And now, excuse me, if you look at the numbers underneath, we're down closer to 5,000 missing, and we've got a strategy for finding many more of those. So it's a pretty exciting project. It takes me a lot of places around the world. I'm not here. I might be in China or Estonia or Madrid or Brazil. In any case, I thank you for being here to hear all this. I hope it all ties together. I wanted to give you a sense of the context and the potential applications of really basic bioinformatics work on proteins and families of proteins, and show you how it's of interest to FDA for new kinds of diagnostic and therapeutic approaches, how it's interesting to us in building this comprehensive approach to the human proteome, hopefully to match the power of a human genome project. Thank you all very much.
You mentioned looking at peptide sequences that were unique for alternative splice variants. And excuse me if it's a very naive question, but how often would you encounter alternative splice variants where, when you're doing MSMS proteomics or mass spec, that you would find that after trypsin digestion, you would not find any unique peptide sequences? And then how would you deal with that problem if it doesn't?
No, this is an excellent question. Question is, in mass spec, since you may apparently know that we don't find all the peptides, how can you solve these questions if the method only detects some of the peptides of interest and therefore some of the proteins of interest? That's why we've switched our strategy to start from the transcripts and go to the proteins now that we have the available methods and colleagues who generate enormous amounts of protein of RNA sequence data. The answer to your question has several elements, but briefly. One of the reasons we do not detect, for the missing proteins, we don't detect the protein, is that we rely on trypsin digestion to release peptides from the protein sequence. Mass spectrometer is most sensitive and accurate for peptides of 7 to 30 amino acids. So if it's smaller than that, it doesn't get picked up reliably. If it's bigger than that, it doesn't fly in the laser stimulated chamber of the mass spectrometer. So that's a pretty special window. That means in the protein sequence, there has to be a site for trypsin cleavage. The trypsin cleavage is chosen because it's specific to lysine and arginine. If there's no lysine or arginine in that sequence of the protein, there will be no tryptic peptides. There could be other kinds of peptides, but not tryptic peptides. That's very important. Also, proteins, some of the missing proteins are membrane proteins, embedded in membranes, hard to release, highly hydrophobic, and sometimes basic. When we look at their sequences, they have these transmembrane domains, which are high structured regions, and in between are disordered regions, which you might be able to cleave rather easily. They're exposed. But those disordered regions typically don't have lysines and arginines. At least the ones that are missing don't. So that's another problem. Or in some other proteins, there are too many lysines and arginines, so they get only these tiny peptides. So this is an inherent problem of the mass spectrometry methodology. And it does lead some people to use alternative enzymes for cleavage. And there are some other strategies that can be used using synthetic peptides to look for matching fragments. Anybody else? Hala. No, it's a wonderful question. Yeah. It's a wonderful question. In fact, I'm one of the founding editorial board members of a new journal called Evolution, Medicine, and Public Health. So, and there are other journals about molecular medicine and evolution and so forth. So it's a pretty interesting question. And what if you've never heard about this before, when I told you that it doesn't exist in microorganisms, not just archaea or some, but essentially none in microorganisms, and a little bit in yeast, different level, and all of a sudden we have this incredibly elaborate spliceosome and function aligned with these various splice variants. It's pretty amazing. Well, one of the important, just an example of an answer to your question, because it triggers many possibilities. One is that we're probably not thinking best about how proteins evolve to think about whole proteins, because proteins have functional domains, binding domains, catalytic domains, regulatory domains. And in fact, it's likely that this whole intron, exon business was a way to identify and maybe segregate these domains and motifs within the linear sequence of a protein. And there's evidence for conservation of domains much greater than of the whole proteins. So I think you're onto something really important. Right. I'm sure that's true. Yeah. Well, absolutely. I think, there are several of you involved with metabolomics analyses. And if we want to understand what makes for different metabolic patterns, you have to take it up a level to the proteins that are enzymes and regulatory molecules for those pathways. And there's already work looking at the transcript level and metabolomics. But the transcript level cannot any longer just be up and down of the whole gene and its whole mixture of products. There's likely to be a lot more information in available data from RNA-seq, trying to match up the different splice variants transcripts with metabolic patterns. And of course, the way to do it is to look at the most divergent patterns for the particular phenomenon and see if you can find an explanation at a higher level in the omics hierarchy. Well, I specifically offered to stay after for the trainees in the PICTP, that's the Proteomics Informatics and Statistics training program. So I'll wait around and see if any of you have any additional questions you'd like to ask in a smaller group. Thank you all very much.